Assalamualaikum, I am Nur Amira binti Ismail. We present an introduction for our research article. The main point of this article is all about one of a critical mental health named schizophrenia. Genetic interaction and environmental factors is the two things that are believed can cause someone to have schizophrenia. So, based on this study, there are two questions that is highlighted to be research questions. First, can DNA methylation, copy number variations of the C4A gene, and higher C4 levels be associated in individuals with schizophrenia? And the second, how does DNA methylation affect the expression of the complement C4A gene in individuals with schizophrenia? To answer the questions, this article comes up with 4 objectives to be achieved. The first one is to examine the DNA methylation levels and the copy number variations of the C4A gene in individuals with schizophrenia and compare it with healthy controls. Second is to explore the association between the DNA methylation levels, copy number variations of the C4A gene, and the higher C4 levels observed in individuals with schizophrenia. The third one is to assess the severity of psychopathology in individuals with schizophrenia using the Positive and Negative Syndrome Scale, known as PANSS. The last one is to determine the potential implications of DNA methylation and copy number variations of the C4A gene as factors contributing to the development or progression of schizophrenia. That's all from me. Then I will pass to the next presenter. Thank you.
My name is Nur Hazika binti Abdurrahman, and I will be explaining about the methodology that was performed in this research. The first step is sample collection. The sample collection starts by collecting 16 ml of venous blood, 2 ml of Buffy-coat that was extracted by centrifugation, and the extraction plasma was sent for C4 level determination. Next, the genomic DNA is extracted and purified from the Buffy-coat in blood using a kit. The quantification and purity assessment of eluted DNA sample were performed. The samples and controls were diluted to 25 nanogram per microliter prior to bisulfate conversion. Then, the C4A primer and probe sequences were designed. The primer obtained was from online tool based on Metalite metal-specific primer design and following the CpG island sequence for prediction. After that, the quantification of the DNA methylation levels of the C4A gene was performed by Metalite quantitative polymerase chain reaction assay. To quantitate, each sample was run in separate single reaction on a 96 well plate. Next, the copy number of C4A from genomic DNA was determined by a droplet digital polymerase chain reaction assay. After the droplet mixture was done, the fluorescence of C4A gene and the amplicons in each droplet was read and the data was analyzed using Quantisoft software. And the last step is statistical analysis. The data was analyzed using various software and various tests was performed and were used where it is applicable in the experiment.
Hi, my name is Nur Inani Amihuda binti Hamri, and today I will present to you the result and the discussion from this study. A total of 183 schizophrenia and 212 health control, or HC, were found towards the end of the research. There were no significant variance in age, gender, ethnicity, or BMI between SAC and HC. However, SAC had a much larger percentage of smokers than HC, along with higher plasma C4 levels. The majority of patients recruited were chronic unstable, as evidenced by a mean illness duration of nearly 13 years and low average PANSS scores. All SAC were also on drugs, the majority of which were atypical antipsychotics. Several studies have previously reported higher plasma C4 levels in schizophrenia, and this study found that the higher C4 levels were positively correlated to C4A PMR values, suggesting that DNA methylation of C4A may indirectly be associated with schizophrenia through high C4 levels. The effect of DNA methylation depends on locus and context. The positive correlation between C4A CCNS and plasma C4 levels correspond with results from other observational human and experimental animal studies. The overall complex variation at the C4 locus may better explain the variation in total C4 levels than C4A CN alone. Despite the positive correlation between C4A and C4 levels, no significant difference in C4A PMR values between SZAC and HC was found. This aligned with findings from two large epigenome-wide studies that detected no differential methylation in or around the C4A levels. The study also found no difference in C4A CNV types between SZAC and HSC, which aligns with previous studies that measured C4A CNS using ddPCR. However, another study that imputed C4 allele variation from single nucleotide polymorphism SNP data reported a positive association between C4A CNS and schizophrenia. But the difference in the mean CN report was small, which is 1.94 versus 2.08. The imputation method has advantage in dealing with the complexity of C4 structural variation and larger sample size, but it was established based on SNP data from largely Caucasian cohorts. Using a binomial logistic regression, the study observed that only C4 levels were significantly related to the risk of schizophrenia. However, both C4A PMR values and C4A CCNS were correlated with C4 levels. Alternatively, the higher C4A PMR values and the C4A CNS could confer higher susceptibility to developing higher C4 in the presence of other factors such as infection or stress, which eventually lead to schizophrenia. The study also found no correlation between PANSS scores and C4A PMR values or C4A CCNS. This lack of correlation with C4A CCNS resembled a previous study involving 271 subjects with first episode psychosis. Measurement of C4A mRNA levels or C4A protein levels will also be more specific than total C4 levels for showing the association with CN and DNA methylation. In conclusion, the study has several limitations, including the need to determine variation within the complex C4 genes, measure C4A mRNA levels or C4A protein levels, and investigate the influence of antipsychotic medication on plasma C4 levels or C4A DNA methylation. Further research is needed to determine the relationship between genetic variations, DNA methylation, and disease risk.
In conclusion, the plasma C4 levels was found significantly higher in schizophrenia cases than in healthy control. There are several positive correlations in several parts of the experiments that my friend has explained earlier. However, there are no differences found in C4A PMR values and no differences of C4A CNV types between these two.